Hello, welcome to another bioinformatics video where I'll be going through how to construct trees and the algorithms that we use to build hierarchical relationships. And we can divide these tree building methods in roughly into two categories. The first are known as character-based methods, and these are based on attributes, discrete attributes. And these can be DNA bases, for example, ATGC, but they can also be morphological features like eye color or behavior. Uh, and the units of change become either like bases in the case of DNA or the discrete categories, the discrete states of these different characters. So for eye color, it would be things like brown, blue, green. And of these character-based methods, the two main ones are one known as maximum parsimony and another called maximum likelihood. And then there are a class of methods based on distance, and distance-based methods are based on the similarity or dissimilarity between pairs of aligned sequences, nucleic acid or protein sequences. And of these methods, there are three types, and in this video, we'll be going through just one of them, this UPGMA method. But first, we'll talk about the parsimony methods. And the principle behind this is it's the idea that the most acceptable explanation is the simplest one, or Occam's razor. And in the context of trees, this is saying that the most parsimonious tree is the one that requires the fewest evolutionary events to explain the sequences that we see. For example, if we have these four sequences here, how might they have been generated? And in the most parsimonious case, we have a tree that is first, we have like the main lineage, right? The initial lineage leading to AAA out here. But then we have it diverging with a single mutation in the middle here. And then that mutating again to give us the third sequence here, GGA. And in the process, we get then two of the results, GGA and AGA, after only one change here and one change here. In contrast, we could have a situation where AAA changes twice. We get two mutations, right, to reach GGA directly from AAA, and then separately get AGA through a different event, get AAA turning into AGA, and then these two events having nothing to do with each other. This is, of course, possible, right? Or plausible, right? It's not impossible for this to occur, but the principle of parsimony is that we accept the one that involves the fewest changes, the simplest explanation. So it is not guaranteed to give us the correct answer, but it's a kind of intuitive or logical rule that one can follow. And then we have distance-based methods, and these are based on the comparison of sequences using what are called distance metrics. And there are different types of distance metrics. The most basic one is called the Hamming distance. And the Hamming distance is simply the number of substitutions that one would need to make one sequence into the other. And for this, you need sequences of equal length. For example, Carolyn here, Catherine here. And in this case, if we wanted to transform this sequence of letters, Carolyn, into the word Catherine, then we would need to execute 3 substitution events for this to work. Likewise, if we wanted to transform this word Carolyn into Kirsten, that would require 3 substitution events as well. And we can do this with any kind of sequence, right? It can be a number as well, a sequence of ones and zeros. and we're simply counting the mismatches basically. And in contrast to the Hamming distance, which is extremely simple and rudimentary, can be calculated very quickly, there's a much more involved algorithm needed to compute something called the Levenshtein distance, but this is a bit more nuanced than the Hamming distance because it takes into account the possibilities of insertions and deletions. So sequences may differ not just by their substitutions or their mismatches, but we could have, for example, an insertion needed to transform the name Ada here into Adam. So that would have a Levenshtein distance of 1. Levenshtein distance also includes the substitution counts. So it's kind of like a more advanced version of Hamming distance. So for Stephen and Stephen here with a PH, we could do one substitution, right, for the V for the P, and one insertion, insert an H here, and that would give us a Levenshtein distance of 2. So it's the minimum number of edits or changes that you would need to make to transform one sequence into another. And for this reason, the Levenshtein distance is sometimes called the edit distance, or more precisely, the Levenshtein distance is a type of distance in the category of distance metrics called edit distances. Then we have a hierarchical clustering technique, and this there are several types of hierarchical clustering techniques, but one of them is called UPGMA, which stands for Unweighted Pair Group Method with Arithmetic Mean. And this is a distance-based method, so it's going to utilize something like the Hamming distance or the Levenshtein distance in order to construct a tree. And what it's going to do is assume the same rate of evolution, so this molecular clock hypothesis applies. The length from the root to each leaf is the same, so it's one of these ultrametric trees. And the logic of these distance-based methods is all the same, namely that the sequences of greater similarity are separated by shorter evolutionary distance. And in this case, this is a way to achieve a parsimonious result. So we start with sequences of equal lengths. And by the way, this content comes from a video on this topic from Oxford University Press. So please check out that video if you want perhaps a better explanation of this. But here we have several sequences, and we align them, right? So they're all the same length. So we put them, we line them all up here in rows. And what we're going to do is go through each pair one by one and count the number of differences. So this is going to give us our Hamming distance. And in this case, we have 9 differences here between A and B. And in contrast to that, 2 differences between A and C. So these different values then, what we're going to do is tabulate them. So we'll put them into a distance matrix. So basically just a matrix here of all the different values. We could fill in these ones here across the diagonal. They would just be a mirror image of these, or we can just fill in these ones to make it easier. And for example, so we said there are 9 differences between A and B, so we put a 9 in this square here. And there were 2 differences between A and C, so there's a 2 in this square. And then you would go through and measure all these distances and put them into this table. And then to construct the tree, you begin by identifying the pairs with the fewest number of differences between them, the smallest Hamming distances. So this pair A and C here has only 2, and this pair B and E here has only 2. So these would form the first links here in our tree, at the bottom of our tree. Then we would go through in the second phase and assemble a new table, which takes into account one of the groups that we've created. So we've created this AC group here, and now we want to compute the Hamming distances of the AC group out to the different members. And this UPGMA method is based on the arithmetic mean. And so what we're going to do is take the average of, let's say we want to get the AC to B distance here. Then we would check the average by checking first A to B, which is 9, and then checking separately C to B. So C, B here, and A, B, they both have 9, so the average is 9 in that case. Let's say we wanted to check D. So we would check first A to D is 4, and then C to D is 5. So we compute the average of 4 and 5, which is 4.5, and that becomes the value we place in this square here. And then we just repeat this for everything, and we fill up the table again, and then repeat this process then for the other branch that we created here for B and E. So you do the exact same thing, compute the average of the distances from B to each element and E to each element, and then averaging them to get the values that you would place in the respective positions. And then once you've done that, we repeat the process of identifying the pair with the fewest number of changes, or the smallest Hamming distance, and then go back to our tree and update it accordingly. So here, AC to D was the winner, so we connect the AC group here to D. And then you just continue this process. We repeat it. Then we create a new table with the new grouping here, the ACD grouping. Repeat the process of computing these averages and fill in the table again. Repeat identifying the smallest distance group and update the tree again accordingly, this time adding the ACD grouping to the BE grouping, so creating this link here. And then finally, you get to the end, and all that's left is F here, which has a tie between these two values here. So the Hamming distance of F to the ACD group is the same as it is to the BE group. So this gives us the final link we need to complete our tree. So to conclude, this is the UPGMA algorithm. It's a distance-based algorithm used to compute a tree. And the idea is you line up the sequences as an ungapped multiple alignment. You compare pairs of sequences to each other to determine how many differences there are. And this is the so-called distance between the sequences. And you construct this distance matrix. And this is simply a table that counts the number of distances for every possible pair. And from this table, you're going to identify the sequences with the fewest differences. And this will be used to assemble your tree. And you would repeat this process until your tree is complete. A modern application of phylogenetics in tree building in modern biology is, of course, the ongoing coronavirus pandemic. So this is March 2021, and as of now, there's a huge phylogenetic tree if you go to nextstrain.org. And they actually, on their website, they have two types of trees. You can check one based on, I believe, based on just genetic changes, evolutionary changes, to look at SARS-CoV-2 and where it fits into the family of coronaviruses. But then we also have this time-based tree here, where we can see on the axis here we have time that is going by. So some of the first people to recognize that the pandemic was going global were researchers at the University of Washington who were looking at phylogenetic data, sequencing data of coronaviruses, and determining, for example, based on these kinds of trees, that the data they were seeing at the time was not simply the result of people who had traveled directly from China, that this had already reached the point where it was spreading from within the population there. And so these were the first warning signs that we were dealing with a truly global pandemic. And then another kind of related topic, also related to tree building, is this idea of constructing multiple sequence alignments. So in previous videos, we talked about how we could use multiple sequence alignments and how they were gave you a lot of information about structure and how we could use different types of profiles and things to represent them, consensus sequences and such. But we didn't talk about actually how to obtain these alignments in the first place. And that's because to know how it's done, it helps to know a bit about how trees are constructed. And one of the fundamental problems with multiple sequence alignment is that it combinatorially it becomes a really difficult problem. So brute force searching for alignments in a brute force strategy does not work. So if we wanted to simply perform a dynamic programming algorithm similar to Smith-Waterman alignment, that would go through and systematically figure out where we need to place gaps to align all the sequences optimally with each other. This quickly becomes very computationally costly. So for 375 residues for 2 sequences, it could take 0.05 seconds. But based on this scaling rule that applies to multiple sequence alignments, a 375 residue sequence times 10 for 10 sequences would take 3.6 days. And then already, if you align 100 or more sequences, we're already up into computation times that exceed the window of what's reasonable. So we need something more efficient, and the kind of basic approach is something called progressive alignment, and this is the idea that we will add sequences to our alignment. So we first perform one alignment, say between 2 sequences, and then we fix those positions and then add a sequence to that. So perform another alignment with those fixed sequences. And the thing to note in this is that the order of sequences can be very important. And because the order is also important, we might attribute particular weights to certain sequences. Some sequences maybe have higher quality data, and we maybe want to align those first because the order that we align the sequences in is going to impact the results we get. So what do I mean by this? So consider the 3 sequences here: GG, DGG, and DGD, and we want to perform a multiple sequence alignment on these 3 sequences. And so let's say we do first DGD with GG. So we perform multiple pairwise alignment on these, and you can see that there are 2 possible places where we could put the GG that are the same, right? There's one mismatch here with the G and the D, and there's one mismatch here with the G and the G and D and the G. So if we're just looking at these first 2 positions here, we could pick either one. But then later on, we introduce this 3rd sequence, DGG, and then all of a sudden it becomes really important where this GG is. If the GG was here, then we would get good alignment, for example, between in this position here. But unfortunately, if we had placed GG in this position, then we get another mismatch here, and then this alignment, maybe this sequence would instead be placed with the GGs underneath here. And so we get a suboptimal alignment as a result of choosing this order instead of this one here. So how do we deal with this problem? The answer is to use guide trees, and the idea is to compare the sequences in a pairwise fashion to identify the most similar sequences, and then we align them in the order of similarity. So how could we do this? So for example, for this DGD example, we would maybe first align DGD and DGG, and then after fixing this alignment, we could align this result with that. So here's the procedure. You first make an initial similarity assessment. So for example, we could use Hamming distance on all our sequences to determine their pairwise similarity. And then from that, we construct one of these guide trees. So we determine, for instance, that red and blue here are more similar to each other. And then yellow then is next most similar to the red and blue group. And then on a separate branch here, we've got green and pink that are related to each other. And then this group is then the least similar to this group. So we construct this guide tree, and then we perform the alignments in this order. So first we start with red and blue here. And then after fixing that alignment, we then check yellow and how it aligns with the fixed red and blue here. And then now we have a group that we can work with in parallel with this, we would try, say, green and pink, fix that alignment, and then merge these two groups together, align these two with each other. So some examples of this in practice are, for example, Clustal omega, and this is part of this EMBL service here. And what you can do is go to Clustal omega and paste in a FASTA sequence, a FASTA file. And in this case, we've used some NGS data, ngsdata.fsa, simply pasted it in. And if we perform this multiple sequence alignment, we can see the results here. So some agreement here in this area, and actually due to these overlap, we see the possibility of maybe getting a consensus from these shorter sequences out here. And then some disagreement down here. So that's multiple sequence alignment, how you get it. And we went over progressive alignment, and this is kind of like the basic thing to understand. So there are other methods for performing multiple sequence alignment. Most of them are kind of similar in some way to progressive alignment. They involve adding sequences and kind of efficiently traversing the space of possibilities in a way that doesn't create too many random accidents. And you have a class that are called iterative methods, which allow you to kind of iteratively realign and kind of refine alignments as you go. There are also machine learning based approaches that are popping up. So this is an active area of research. These are some softwares, for example, Clustal Omega we mentioned, but then also you have things like Muscle and MAFT. These are coded in compiled languages, so they run quite fast. And because multiple sequence alignment is so computationally intensive, involves these multiple pairwise alignments, you have usually a focus on performance. So here we can see a table comparing the total computation time for these different techniques. And then a final note, just some recommended reading on the topic of trees and hierarchy. I think personally that this topic is very related to the idea of computation and the relationship between computation and biology. So we see a lot of hierarchical structures occurring in things like operating systems or computational programs have hierarchical structure. And we see hierarchical structures emerging in biology. And they have some things in common, right? In computation and also in biological systems, you tend to have input signals, output responses, usually a purpose of some kind driving the input to output, and a chain of cause and effect in between them. And this book here by John Mayfield, I think, is really good. It's making a case for evolution as a type of computation, really taking a computational perspective to the topic of biology as a whole. And then somewhat related to this is this book called The Book of Why by Judea Pearl. And he's a mathematician who developed this framework for understanding cause and effect in a rigorous mathematical way, probabilistic way. And I think it's a really interesting way to think about things that happen, chains of events, and kind of universal way to tie things that maybe on the surface don't seem related to each other, like computation and biology, but which maybe share something deep at their core, and that is creating these hierarchical structures. So just some food for thought. And that's all for this video. So I'll see you in the next one.